autologous T cell therapies, gene-edited T cell therapies for individual patients in and of itself is hugely disruptive. You have to apheresis the patient, ship the apheresis. In our case, we freeze it, and then thaw it, and then put the cells into a manufacturing run. You add in a lentiviral vector that's got the T cell receptor gene constructs in, grow the cells, freeze them down, undertake the release assays, and then ship it back. So that whole vein to vein time, which is really crucial from the patient's perspective, is challenging in and of itself. And I think one of the things we did early on after raising money, taking the company public, was to invest in our own manufacturing capabilities, both for the cells, which we make in our facility in Philadelphia in the Navy Yard, and also making lentiviral vector, because many of those in this field will realize that, or will know that there are very few external suppliers who can provide vector, GMP grade vector, and a timetable when you actually want to have it. So that's become quite a bottleneck around the field. So we invested in our own capabilities to do both of those things, so that we had a lot more control over quality and timeliness of supply, scheduling of patients, and it's also allowed us to bring improvements into the process. So in terms of the vessels that you grow the cells in, the agents that you use to stimulate the cells to grow, as well as the vector and the copy, the sort of titer of that vector. There are all sorts of elements of actually making the cells which are challenging, which we've worked hard to optimize and improve. The timeliness of getting it back to the patient, being able to freeze on both ends is really important. Getting into Europe has been, is challenging. We were actually making cells in the US and shipping them. You have to have a qualified person in Europe to release the cells. So we've now got our own qualified person inside the company to do that. So there are sort of everywhere you look, there are challenges. I think also, perhaps even stepping back beyond that, in the early days, most of the clinical sites that had experience of T cell therapy were in the hematological setting, and obviously where the two marketed products are out there, probably soon to be more at the moment. We were actually building a footprint of clinical sites in solid tumours, where the idea of taking an apheresis, booking a slot, shipping for manufacture of cells, the whole safety profile, the screening, etc. Apologies, my computer was all new. So we've now got in excess of 30 sites, and we've, as I mentioned, we're in Europe as well. We're in Spain and France, as well as the UK. We're in Canada, and I think we've had to put an awful lot of time and effort into building up the infrastructure, as well as maintaining the dialogue. We've had a few safety issues, predominantly related to the lymphodepleting chemotherapy you give the patients prior to them receiving the cells back, which effectively knocks out their endogenous T cells and creates space for their incoming T cell product to engraft in the patient and expand. But those can have toxicities. So dealing with late stage older patients who are less able to tolerate that chemotherapy before they get the cells has been a challenge, and I think we spent time trying to make sure that we have letted out in the right place on that. So I could go on and on. The screening, diagnostic, screening time, getting HLA tested, getting an antigen, so getting a biopsy sample to be able to screen for the antigen in the first place. There's quite a lot. And then I think where we are now is getting samples back from patients that enable us to understand those that are responding, why they're responding, those that don't, why they don't. And so that's probably where, we're very laser focused right now to improve upon what we've seen and to follow to chase down the responses towards, marketable, developable products.